Hello and welcome back. In this next video, which is entitled Meiosis Express, I want to talk about the cell divisions which result in the formation of our gamete cells. That is, our sperm and our ova cells. All right, so before we even begin to go through this complicated diagram, I want to explain a little bit about the genomic content of our cells. All right, so our somatic or our body cells possess 46 chromosomes. And let's just write this little note for ourselves on the back of the sheet. All right, so somatic cells, these are the body cells, and these are all the cells that are not the sperm and ova. All right, we can say that they have a diploid number of chromosomes, and we can assign a number to this set of chromosomes. 2n equals 46. All right, so where does this n come from? Well, this n term refers to the number of chromosomes in a set of chromosomes. All right, so by contrast, our gamete cells, these are the sperm and ova, these are haploid cells. All right. Now you guys know that sperm and ova, we inherit the DNA, we inherit DNA via sperm and via ova from our mother and our father. These cells can also be assigned a number of 1n equals 23. So there are 23 chromosomes in a sperm cell and 23 chromosomes in an ovum or an egg cell. And when those combine, we get a single fertilized cell called a diploid zygote that has 2n equals 46 chromosomes, and that will be your genetic makeup for your body cells or your somatic cells for the rest of your life. All right? Now, the goal of Meiosis Express is to take one of these diploid somatic body cells and go through a round of cell division, not once, but twice, to result in 4 haploid gamete cells. So let's write this down. The goal of meiosis is to undergo 2 divisions to result in 4 haploid gamete cells. All right? Now, not only do we result in 4 haploid gamete cells, but these cells are genetically distinct from the original diploid cell from which they came. These are genetically distinct from the parent's cells. All right? And we will talk about the shuffling events which result in kind of like a shuffling of the chromosomes that enable us to pass on a diverse combination of genes and chromosomes to our offspring. Okay, so let's take a look at this. Let's first label each of these stages because they very closely resemble mitosis. All right, so let's make this simple for us. This is prophase 1. All right, so just like in mitosis, we have a prophase, prophase 1. Then we have metaphase 1. After metaphase, we have anaphase 1. And then we have telophase and cytokinesis 1. Okay? Now, after telophase and cytokinesis 1, we go back to prophase, but this is a second round of cell divisions. This is prophase 2, which I'll just abbreviate P2. Metaphase 2. Anaphase 2. And telophase and cytokinesis 2. Okay, so let's discuss the events which are unique to meiosis. So we'll talk about the similarities and the differences as we go through this diagram. All right, so just as in mitosis, we have a prophase of meiosis. And in prophase 1, all right, the nuclear envelope disappears. Sorry, that says disappears. You can say disappears or dissolves, all right? And then we get all 23 pairs of chromosomes coming into very close proximity with each other, all right? Now, we have these chromosomes that condense, and the 23 pairs align. All right? And when they do so, they come into very close proximity and crossing over occurs. All right? This is a major event that occurs during prophase 1. And I want you guys to go to the video entitled Crossing Over that explains what the significance of this event is. Basically, it allows us to take the chromosomes from mom and the chromosomes from dad, and we can swap out arms of these chromosomes so that when we pass on these chromosomes to our offspring via the sperm and the ova, they contain unique combinations of pieces of chromosomes from mom and pieces of chromosomes from dad. All right? All right, moving on to metaphase 1. All right, what is the similarity here between metaphase 1 of meiosis and metaphase 1 of mitosis, or metaphase of mitosis? Well, we get the chromosomes lining up, they meet in the middle. However, we do not have the centromeres down this imaginary metaphase equator. Rather, the difference here is that the homologous chromosomes randomly align on either side of the metaphase equator. Okay? So rather than straight down the middle, we get the homologous chromosomes, 23 pairs aligning on either side. All right? Now this stage right here, we can think of like a shuffling. All right? So not all of the chromosomes from mom go down this side, and not all the chromosomes from dad go down on this side. All right? It is a shuffling. So some of moms and some of dads, some of moms and some of dads. And how many combinations of dad's chromosomes with mom's chromosomes line up on either side? Well, that can be expressed as 2 to the 23 number of combinations. And that is more than 8 million unique combinations of shuffling mom's and dad's chromosomes on either side. All right? So here we have crossing over as a shuffling event, and we have random alignment as a shuffling event in metaphase 1. Okay, in anaphase 1, right? It looks like anaphase of mitosis where we get the separation of these, but remember in mitosis, we split the sister chromatids. Here, we do not split the sister chromatids yet. Rather, we segregate the homologous chromosomes. So the major event which happens here is we segregate the homologous duplicated chromosomes. Right? This is where they're split apart. And in telophase and cytokinesis, each one of these cells is going to have 23, let's see, are they duplicated or unduplicated? Duplicated chromosomes, all right? We haven't split the sister chromatids yet, all right? Now we go on, we take these two cells, they undergo telophase and cytokinesis, and now we have two cells, each with 23 duplicated chromosomes. We go through meiosis 2, okay? Here, this is what looks most similar to mitosis, right? Except rather than 46 chromosomes aligning along the metaphase equator at their centromeres, 23 duplicated chromosomes meet in the middle. They align at the metaphase plate or equator. And in anaphase 2, 23 duplicated chromosomes are split. So here we can write, we split the sister chromatids. All right? So in anaphase 1, we segregate the homologous duplicated chromosomes. And in anaphase 2, we finally split those sister chromatids. And in telophase and cytokinesis 2, all right, now each of these 4 haploid gamete cells will have a complement of 1n equals 23 chromosomes. All right? And these cells will go on to become the sperm or the ova, that when the sperm fertilizes the ova and vice versa, the resulting zygote will have 46 homologous chromosomes. In other words, 23 pairs of homologous chromosomes. All right? So now I'd like you guys to go make sure that you understand mitosis. It will help you understand the events that occur in meiosis. Make sure you go watch the video that explains crossing over. All right? Now, all of this is going to lead into our chapter on genetics and what implications there are for crossing over and random alignment. Okay, until next time.